As you can see on the screen, oral drug discovery has been the preferred way to discover new molecular entities. But looking at the statistics here, it is quite clear that further innovation is not possible unless we look at other macromolecules and peptides. How did we go from peptides being pariahs to hopefuls? Well, as we said, only looking at oral drugs limits the rate of discovery and innovation. We now have a lot of research and applications behind alternative routes of administration. There is greater acceptability of these routes for patients, and any of the insurmountable problems like low bioavailability, rapid metabolism, and thus limited half-life are being overcome. There have been lots of examples that have been identified as bioactive peptides. More than 60 to 80 have now been approved by the FDA for various uses, and thus they are worth our attention. They do have certain advantages as compared to larger macromolecules like proteins and antibodies, in that they have better tissue penetration, they elucidate a lesser immune response, and they have a lower manufacturing cost. They have other advantages as compared to small molecules, in that they have better selectivity, specificity, and efficacy. It's also seen that they have less systemic toxicity as the peptides are rapidly disintegrated or metabolized into their constituent amino acids. And like small molecules, they are also amenable to SAR. There are some rules that are available in literature to design bioactive peptides, and these are some rules that can be looked at. But rather than use specific rules, why not use deep neural networks and GANs to generate bioactive peptides? So the subject of this challenge is to use a generative adversarial network, which is composed of two networks, model A and model B. Or as you can see, there are two neural networks on the right. The generative model or model A takes in fake examples and generates more fake examples. And the discriminator network takes these fake examples, and that discriminator network is a normal classifier which has binary classification, and it has been trained with labeled examples of real bioactive peptides and false bioactive peptides. Now you have a lot of freedom in trying to understand what is the non-bioactive data set that you will choose. It could be if you choose bioactive antiviral peptides, there could be other bioactive peptides as a negative set, or you could choose more random form of peptides or peptides that do not follow the rules that were shown on the previous slide. So there are variety of ways in which to generate the negative set to train the discriminator network. And the generator network can also take in some of these fake peptides or peptides that have a similar composition, amino acid composition to real bioactive peptides, and that can be the training set for the generator network. So over a few iterations or epochs, the network, which is a form of supervised learning, actually has an evolutionary competition and learns to discriminate between real bioactive peptides and false ones. But in the process, it also generates a lot of novelty. So the hope is that by using this kind of a network and some tweaks like in the network topology, the parameters used, the negative sets used, and several other things, or combining that with the rules shown earlier, you will come up with a framework in Python or in R if you prefer that, using GANs um, to generate biologically active peptides. The most preferred way to generate these networks is probably PyTorch. Thank you.